Welcome back. We are still talking about predator-prey interactions, a little bit more on sort of the evolution of avoidance, escape, things like that, by the prey, so that they don't get eaten by the predators. And today I want to start with a quick sort of diving into the idea of coevolution. We're going to get into that in more detail during the lecture, but I want to make sure that we're all on the same page, that we're actually talking about a very tight interaction between two species. They are affecting each other's evolution, coevolution. So it's often confused with convergence, convergent evolution. Convergent evolution is not coevolution, or coevolution is not convergent evolution, because the two species are not interacting. So in this image here, there's a hornbill from Thailand. Looks very much like a toucan from South America. They both have a very similar habit of eating big seeds and using their bills as sort of ways of getting food. And they're both birds, okay, but they have had no interaction with each other. They've just converged on that bill type. And so that's very different than coevolution, where you would have two species that have been playing off of each other and they've had a coevolutionary story. The classic example, I suppose, could be coronavirus, where initially some coronaviruses have been around for a long time, and we have, in the human population, we're immune to those. But occasionally one evolves that is a whopper, such as COVID, coronavirus 2019, or infectious disease, ID, COVID. And that one, it evolved to override our resistance to the typical coronavirus. It's wiping out large segments of the human population. Those of us that remain will be resistant, and eventually it'll kind of fade away. That's coevolution. Eventually we become resistant. Eventually it evolves a new version that's virulent, and then we become resistant, and then there's a virulent strain, and then we're resistant. Okay, we'll look into a little bit more detail on that. But that's coevolution, classic. Convergence, very different, two different continents. There's no way these two species have interacted in that fashion at all. They're just on different paths. They just wind up looking the same. Another example, cacti in North America, and euphorbia, like the crown of thorn cactus from Africa. Okay, very different plants, but we often put them in the same group if we're not careful. All right, so let's look at another example of coevolution. This is monarch butterflies and milkweeds, and fantastic story. So you know about these monarchs. You know that they're having trouble. Actually, this year they did very well, as it turns out. Hopefully enough people are planting milkweed to help these guys make their way down to the breeding grounds in Mexico and back. But they need a lot of food to do so. They need their milkweed. So they are getting nectar from the milkweed, but they're also getting a toxin from the milkweed that prevents animals from eating them, birds. It doesn't prevent, it discourages. And they're brightly colored, so the birds recognize them and say, yep, nope. There's another one called a viceroy, by the way, that looks very similar but is not toxic. That's the mimic. Is that Mullerian or Batesian? Go back and look at the last segment and tell me. Okay. The caterpillars are eating the leaves. So these guys drop their eggs on the leaves, and the caterpillars come out and start chewing on the leaves and get that sap, which is toxic to most insects. Works really well for most insects. For the plant to get rid of most insects, herbivores. But this monarch has figured out a way to override it. I'll show you another example in a minute of how that might evolve. But birds that eat that caterpillar learn very quickly that that's not food. if we go back to the picture, note how brightly colored that caterpillar is. You can see it a mile away. So can the bird, and the bird says, nope, looks like I need to keep looking for some food somewhere else. Here's another example of the same thing. This is Heliconius, beautiful butterfly up here, and the passiflora, or passion flower, and that Heliconius gets its protection from the plant. This one, right? Yeah. So, and then you can see these spikes. When you see a caterpillar like that in the tropics, don't touch it. More than likely you're going to get something that you didn't want. That tells you there's something wrong with that caterpillar. Right for the caterpillar, it's avoiding predation, but not good for you to touch. Oops. And then this one, another coevolutionary story, and this is the one where I can actually talk a little bit more about the actual development of the coevolution. So this is a swallowtail butterfly caterpillar, brightly colored, right? Birds, don't eat me, bad idea. With Queen Anne's lace. Now Queen Anne's lace belongs to it's in the carrot family, right? We eat carrots. If you dig up a Queen Anne's lace, you'll get what looks like a carrot root at the bottom. In the same family as poison hemlock. Don't go around eating things that look like carrots if you don't know what you're doing, because you may be eating something that's toxic to you. Poison hemlock is strong enough to kill a human. In fact, I think that's how they killed Socrates, when he pushed his luck a little too much. But these guys, the Queen Anne lace, not that toxic, but toxic. And the caterpillar eats it, gains that toxicity, and it helps protect it from birds. How did that happen? The first caterpillar that ate Queen Anne's lace of the swallowtail group probably didn't do so well, but survived. And as time went on, more and more survived, and eventually you get what we have today, a very toxic and resistant caterpillar, right? Same with the plants. And so there's been some wonderful work on this group, both the plants and the caterpillars. And basically this is from Barenbaum from 1983, not that long ago. In an earlier slide, I talked about some of the chemicals that are produced. Bear with me, I will hopefully, flavonoids. So coumarin is a type of flavonoid. Sometimes they call these things secondary metabolites because they think the plants just have so much energy from all that photosynthesis, they don't know what to do with it, and they start just cranking stuff out. Maybe then it winds up having a use down the road. I don't know. I don't want to go down that path. But coumarins are flavonoids, and the early coumaric acid is not so toxic. So the species that have this, caterpillars can eat them and it's not so bad. Okay. Note that this same chemical then can be fed to a different metabolic pathway and become part of the lignin formation of plants. Or it could go down this pathway and become this umbelliferone pathway, which is toxic. You have nice ring structures. That's always a warning sign. Those alkaloids, that is the caffeine, the tobacco and such, those also are ring structures. So when you see that in a plant, you can bet that that's probably something that is a defensive chemical. And so early plants that had this path, not so toxic, but as you go to the right, you become more and more toxic, and fewer plants can do it, can make that particular compound. And so by the time you get to the Queen Anne's lace, it's a nasty chemical, or poison hemlock. It's bad. In fact, it can kill you. So the early caterpillars can handle the early versions, but by the time you get out here, the caterpillars that could live on the plants that make this stuff will be quickly killed killed on Queen Anne's lace. They no longer can survive. So my point here is this is coevolution, right? The plant has a bit of a toxin. Caterpillars are eating it at will somewhat. Some things aren't able to handle it. But the plant then evolves a more strong version of that chemical, and now fewer caterpillars can survive it. So the plant makes an even stronger version, and now we get to eventually to the Queen Anne lace, and only swallowtail caterpillars can survive. So classic coevolution story. Okay, here's another one. It's a rabbit. It's just a little rabbit. And the other guy said, well, anybody recognize what I'm suggesting here? Nee. It's just a rabbit. And the guy goes into the cave and comes out all chewed up from that rabbit. That rabbit looks pissed. That's why I devolve into Monty Python. I guess sometimes rabbits aren't really happy to have their pictures taken. This is a story about rabbits in Australia, not in England. Apologize for the reference to the boys in Monty Python. Your book highlights this story where there's this disease of rabbits called it's a myxovirus. And when they first introduced it, so rabbits had been let go in Australia, another classic oops, and they overran Australia and started just eating everything. And they said, well, we have to control this horrible pest, if you will, in Australia. And so someone said, you know what, there's this there's virus in, at the home range of our rabbits that we can now introduce into Australia, and maybe it'll knock them down because it's pretty virulent. So they go and add the first several rounds of that virus, and it's an epidemic. It kills nearly 100% of the infected rabbits. Okay, so real virulent virus. Guess where the word virulent comes from? A few of the rabbits survive. So in the next round, you start getting more rabbits surviving that virus. Okay, and then in the next round, more rabbits, fewer are dying, right? So we're going down in mortality, up in survivability for those rabbits in the face of this virulent virus. And so by the time you get to the sixth round, the rabbits are hardly noticing it. Okay, a little over 25% are dying, but nowhere near the close to 100% at the beginning. So they now, like the cactus moth story that we talked about some time ago, they now go in and occasionally release a new strain of the virus to keep the rabbits in check in a very careful approach. This, by the way, is not unlike any disease, and we're talking about COVID these days. Watch the storyline on COVID. You're going to see that eventually humans are going to be less affected by it, particularly if we start getting smart, have a nice vaccine. But it would happen anyway without the vaccine, because the vaccine is basically training our immune system to resist coronavirus. But we're also training that system just by exposure to the coronavirus in nature. Unfortunately, many people die in the process, but a few survive, just like that virus rabbit story. This has happened with humans over and over again. And I think the more fascinating story on that would be my understanding is syphilis was picked up by early colonizers in North America, European colonizers. They brought it back to Europe, and it was one of the plagues. It killed off enough people. I think COVID could be considered a plague down the road. But it killed off enough people, and it killed them quickly. It killed them so quickly they didn't have time to undergo further sexual reproduction and spread of the syphilis. So over time, syphilis became less and less mortal, or I should not mortal, killed people slower, just put it that way. And sort of became benign, went into remission, got into the brain, causing dementia, got into the heart, I believe. And as the delay for mortality increased, there was greater chance for spreading the disease. So good for the syphilis, bad for the humans. So there's a coevolution going on there in terms of when the actual organism dies when it gets infected by syphilis. Okay? Similar to what these rabbits were doing, I'm sure. All right. Now, we do have problems like this in our crops, managing genetic resistance in wheat to a rust pathogen. So that picture in the lower right is a rust on grass, Puccinia graminis, or wheat is a grass, and it gets into the leaves and then eventually bursts out and makes all these orange, rusty-colored spores. These what are called, I love this lecture because I get to use big fancy words, what are called pustules. Now, this is a problem for our wheat production, and it turns out, though, that the USDA agricultural system and farmers are very careful with which plant strain they plant in the face of the virulent strains of the rust that are in the area. Okay? And so depending on what year you look at, and here's a few years from 1956 through 1968, different strains of the rust, the virulent strains, were around. Sometimes, like 17 was a really big problem from 56 until about 66 or almost through 68. But you can see it's became, the wheat plants became more resistant towards the end of the 17 reign, if you will. 15, 9, 11 were around. 18 wasn't even present. It shows up in the early 60s and becomes a huge problem because there's no resistance. The plants hadn't been exposed to it. So that coevolution hadn't happened. The coevolution did happen with 17. That's kind of just, okay, yeah, so what? Interesting. But actually now we use this information in our planting strategies every year in our wheat crops. That should have come out in a couple different patches. But in this hypothetical example, farmers are given a resistant cultivar to plant, and they do, from the southern U.S. all the way up into Saskatchewan. All right, they can't just go out and plant anything. Although there's always a battle between, somewhat of a battle between organic and commercial or standard farming, because the organic farmers can often get around this. They don't want to be given what plant to plant. They want organic conditions. Anyway, so initially they are given cultivars that are virulent to whatever virulence genes are present from last year. They're still around. Those rust strains are still around. So they have to plant the next year's crops resistant to what was present the previous year. Okay? And eventually the rust overrides that resistance. And so you just have to stagger your cultivars in the face of the rust evolution. The rust evolution is rapid, very rapid. Fortunately, we can manipulate the wheat evolution in the greenhouse to evolve new resistant strains just by interbreeding cultivars. So for instance, early in the season in March, they might plant a resistant strain A or a cultivar of wheat resistant to strain A, which is prominent in the southern United States that year. But by the end of the season, they have to stack in a second resistant strain or resistance to a second rust strain. So we started with A. By May, and I'm making this up, but basically it's the same idea. By May, you have cultivars that are resistant to both A and B. By July, you don't have to worry about A anymore because you've basically gotten it out of the system, but now B is being overridden by, the resistance to B is overridden by the rusts, and now we're adding yet another strain resistance to rust strain C on up. So this is actually an international pattern because as the season progresses north, you're given different cultivars to plant in your particular area for a variety of reasons. Some of it has to do with how they can grow in that climate, but also resistance to the local pathogens. Okay, and I want to touch, unfortunately for you guys, I suppose, my PhD is in plant pathology. That's why you just got that little lecture just now on resistance. And I want to end this lecture with a quick touch on two different kinds of resistance that plants, we can use plants as the best example, have. The first is horizontal resistance, and something like bark is a good example. It's not specific to anything. So if a bacterium lands on bark, it doesn't get in. If an insect lands on bark, unless it's figured out a way to chew into it, it doesn't get in. If a fungus lands on the bark, unless it's figured out a way to get around the resistance of that bark, whatever, figured out a way to chew it up and get in, it doesn't get in. That bark is a really good phenotype against things getting in. Your skin is a really good phenotype. Most things don't get across your skin barrier because it's resistant. And it's a horizontal resistance. It's a low level of resistance to all sorts of things that might try to get in, right? So not particularly strong resistance, but it works in general, and it has multiple genes. They're always being produced, so they're constitutive. You always have that barrier, that resistance, and you don't have to turn it on and spend energy turning it on. It's just a low input of energy as a resistance. But if anything overrides it, you're toast. You're in trouble. So then we switch to something called vertical resistance. And now it's usually one pathogen, one plant. An analogy would be the milkweed toxin and the caterpillar, right? The caterpillar figured it out through evolutionary time. It's no longer affected by that particular substance. And it's usually one-to-one, okay? In this case, one rust on one plant. If you put a grass rust on the wrong grass species, it won't be able to survive. Put it on wheat, does fine. Put it on corn, maybe, okay, another grass. So anyway, it's usually highly specific, one pathogen against one plant. It is induced, so often the plant won't turn that particular resistance strategy on until it recognizes the pathogen is around. It might recognize something from the pathogen, a bit of its protein, a bit of its chitin if it's a fungus or an insect. Or it can recognize that its own cells have been damaged. So the insect, for instance, has chewed into the leaf, and that releases, various polymers out of the cell, and then the plant says, whoop, something is busting into us. And you can mimic this by just clipping the leaves and see that resistance pathway initiate. It starts a resistance pathway, a cascade of, that should have only been one. Let me see if I can go back to just one. Nope, it's going to do both. Starts a cascade of events causing plant cell death, but in addition to the cells of the plant, the insect, if it's embedded, or the fungus, or the bacterium, dies with the cells. And you see this all the time as brown spots on leaves. If you just go out, especially in the fall, the leaves are dropping, pull up those leaves, and you're going to see all sorts of dead necrotic spots because the plant has undergone this sort of cascade of cell death, walling off a pathogen and keeping itself safe that way. You and I can't do that as well. We're not sort of free to just like kill off parts of our skin. It happens, but it's really not the best option for us. We have other immunity pathways. The specificity here develops just like I've described because the pathogen overrides the resistance. Maybe it stops producing the chemical that was recognized by the plant, so the plant stops recognizing it, and now the pathogen can do its thing. But then the plant will then figure something else to recognize and becomes resistant again, and you get into that genetic arms race. Sometimes called the Red Queen hypothesis. All the species are running just as fast as they can just to keep up. But that's another topic, maybe an entire PhD for yours truly. Okay, that's the end of chapter 14. No, there's one more segment that I'm going to get into, And I'm going to dig into a couple of interesting stories that you know about. The chytrid fungus on frogs. You know a lot about that, but I'm going to talk to you quite a bit more about it. It's a fungus, sorry, that's what I'm a mycologist. As well as what other organisms are getting hit these days by the different chytrid fungi, what we can do about that. And then get into a little bit on other pathogens that are affecting things like snakes now, etc. So we'll see you next time. That's it for this segment. Have a good session, wherever you're going. Enjoy. Cheers.